Okay, I think I can slowly then start this small session. So welcome everyone. It's a pleasure to have you here. I will first quickly share two links in the chat. So the first link, let me know if it's available to you, should contain the slides that I'm going to show today. And the second one is the link to the repository where the plugin is currently hosted. So this is also going to be in the slides, but just so you have it already. And now I will try to share my screen to the presentation. And since while you're muted, I cannot hear and or see you well during the slides, please unmute yourself and let me know at first if you cannot see the slides, and if you have any questions, feel free to also raise them during the talk or after it. Then let me put this on presentation mode. Okay, so my name is Marcelo. I'm a postdoc and I'm working here in Dresden, Germany, and I work in the bioimage analysis technology development group from the Physics of Life. And today I would like to present this plugin, which I recently released, which is called napari-signal-selector. So it's a plugin to interactively annotate signals from object features in napari. So at first, if these terms like signals or features is not very familiar or clear, I would like to start with a brief general definition of these things. So maybe it becomes more clear depending on the audience I have. We could say that features are individual measurable characteristics or properties of objects. So imagine in your images, you have some segmented objects, and then you could measure some features or properties like area, their intensity, their average intensity, and many others. And signals can be defined as a meaningful patterns or trends in data that convey some information. So what we can relate from these two is that if the objects are uniquely identified across an independent variable, usually time, then signals can be thought of these object properties when the order matters. And this is maybe still look vague, so I brought in you a small example. So again, you have some, let's say, segmented nuclei or objects. And then, of course, here is an example where you could uniquely label them and measure some features or properties. And then if you plot this on a graph, you could see overall like a distribution of intensities, for example. In this kind of graph, it doesn't make much sense to connect them by line, at least not most of the times, because these labels may not be related one to each other. So a line plot like this doesn't usually make much sense. On the other hand, if you have measured something like over time, then you usually want to connect those. So this would be, and of course your x-axis would now be the frame or the time information. And then you could measure these features, in this case, over the frame number or over time. And after that, you could, in theory, be also interested in calculate some other features from these signals, what I'm now calling, like this would be, so if this is like mean intensity over time, then if you get the average of this, it would be like mean intensity, a bit funny, or some people call it grand mean if you do the average of the mean. Then it makes sense at least to track them over time in this case. And signals in microscopy images, I brought some general examples I could think on top of my head. So they are very often over time. So this intensity over time, area over time, position over time, or I don't know, let's say the measure angle of the object over time and velocity over time. But keep in mind, it's not always the case. So I could think about a couple of examples. Depending on your daily work, you can know others, but you could also measure the intensity over some space like if you have some objects and you want to measure the average intensity over different radii or over different angles, for example, that would essentially be signals, but over different places in space. And then you could also have, like, in some cases where you can have multi-channel hyperspectral data, then you could have the same, let's say, intensity over different wavelengths. So that could also be a different example. But then my creativity stopped a bit, and usually time is what most people relate to. Since we're talking about signals, I think now the definition should be a bit more clear, at least how I usually see them. I have a background as an electrical engineer, so that's maybe why I always like to determine them like that. I was looking in ways that we could plot them basically using Napari, which is a nice software for visualizing images. And I found many plugins already that have some sort of plotting capability in them. So there's like already a Napari plugin which does time series analysis called Napari time series plotter. There is Napari plots, another plugin I found where you basically can turn the Napari canvas into your full plot. There are others, like with the Napari clusters plotter, also developed by our group, where you can do unsupervised machine learning and do these selections and see the results back. And there are these others. There's also the Flink Phaser plotter, which I recently released. And to be honest, the plotter is basically based on the clusters plotter, so it's not necessarily a new plotter. And one that got my interest from the very beginning is the napari-matplotlib. So this is also an Napari plugin, which is being developed and maintained by matplotlib developers. And if you don't know matplotlib, so it's one of the most traditional libraries for plotting in Python. So I have been in contact via issues in GitHub with their developers to actually see how I could also build on top of that. And that's what I have done so far. And then I came with this napari-signal-selector plotter. So the plotter itself is built with the napari-matplotlib plotter with a few modifications, as you will notice. And the idea here is that assuming you already have like a result of a segmentation and you have measured some features of these objects. So in this case, of course, over time. Of course, this is a synthetic image. I'm sorry, I don't have a more biologically related sample here, but let's quickly imagine this as being different cells or nuclei or objects with different shapes, and they have different intensities over time. So with this plugin, you would be able to, by selecting from these fields, maybe sort their signals by their ID, which in this case is the label number, and then you could plot them to the side of the Napari main canvas, and you could relate like the colors to the original objects. So this example I mentioned, it's provided as for now the single sample data from the plugin. And as you can see, once you open it, it generates two layers in Napari. This one that you see is like the time lapse, so it would be like the grayscale that changes over time. And the second one is just the result of the very straightforward segmentation of these objects. And then, of course, once you have it, then we reach this previous slide where you could select what to plot against what. And what do you need in order to have, in order to be able to do that? So this plugin expects at least these two things. So the first one is it works with an Napari labels layer. So the labels layer is usually the one used for showing the results of the segmentation, as you can see from this example to the left. And it also expects that you have measured some features of these objects. And so it expects a table like this one to the right. So it should have like a label column or any identification of your objects column that should relate to the labels in the labels layer. And then other features that could be something like the time and maybe intensity, as I said. So this label column is important that it should relate to the label numbers in the labels layer. And you can also check that by looking at as the label field that shows up once the labels layer in napari is selected. By code, so if you have some coding skills, which you may have, I don't know, it should be just that. If you have that structure and then you add the labels layer like this, then it should be good to go. And then I'm going to go over some tools. So the idea here is not only plotting, but being able to actually interact with these signals. So we have, I have programmed here this first, so these are part of this traditional matplotlib toolbar on the top. And then beyond that, I added a few extra buttons. So the first one is this selection tool. So this will actually allows you to individually select and highlight some signals to further manipulate them later. And then you could do that by clicking over them. And if it's too crowded, you can always, for example, use the zoom tool from the traditional toolbar and then look closer and then be able to better click or select your signal. Another option would be like to use, for example, here I'm using the picker tool from the labels layer. So you can pick, like click on top of one label. And then if you click on show select, then it will in both the Napari canvas. So this is native Napari. I did not do anything there, but I just link it back to the plotter. So you see only that particular object that you have clicked, and then you can click on it to have it selected. Further than selecting, you can now provide some annotations. So in Napari, you could already do that by the labels layer. You could draw over your labels in a new labels layer and then provide annotations to your objects. Now, you could also now provide annotations to your signals. So after you have selected them, you can go to the signal class small spin box, and then choose a color or a number, and then click. And the signals that were selected will be now annotated with a different signal class. And maybe you can already see where this can be going. Like you could annotate your signals to further provide these to some sort of machine learning classifier. And that's the late goal of this plugin. But this is only for the annotation tools. Always a hint here on the bottom, so I don't forget as well, you don't forget if you use it, don't forget to right click to unselect previous signals, because the annotation may be drawn on top. And then I want to get feedback of how the visualization looks good. So after you annotate them, maybe seeing that they're still selected is not very clear. So for now, if you right click, you unselect whatever was selected, and then you don't misannotate. So I've also introduced this option to maybe select sub-regions of the signal. So if the signal one or more is selected with the selection tool, you can use the span selection tool to actually select parts of the signal. And if you hold shift, you can select like multiple parts of the same signal. And of course, then you could then provide annotations to just parts of the signal. And these signals are periodic, so of course they repeat, but you can imagine maybe different cell activity, and then for some reason, if you want to have an annotation on that particular part, you could have it. So here I show how to annotate with two different signal classes in different regions, the same signal. Of course, you will make some mistakes, and then there is a delete option. Here I'm zooming in to better be able to select. So let's say I didn't want the signal be annotated as class 2, and then you can select and then click on delete, or just annotate it with class 0. And you can always start over. If you hold shift and click anywhere, it will select all the signals, and then you can delete by clicking on the trash can as well. So these annotations are, of course, saved back in the table. And then if you want to see the table, you can use the show table menu from another plugin, which is a dependency of this one, which is napari second image region props developed by Hot Robert. And you can basically visualize the table back in napari. And there you can see that a new column gets created, in this case called annotations. And then that's how you can have access to the table. And also you see on the top right, you can save this table as a CSV to further do your analysis elsewhere if you like. And there is this option also, if you click on show selected, you can see the row on the table corresponding to the label in the image. This was already from napari schematic region props, but you can also then in the plotter, of course, see the same thing in case you really want to dive into the data. So that's currently what it's capable of doing. And then I'm going to show a couple of slides how I briefly see this being used. So the idea here is to first release this alpha version and collect some feedback. So again, thanks for joining this session. So I hope that you give it a try. And I just released this alpha version yesterday or before yesterday. Let me know if something's not working. It's very important that they get feedback so I can fix and then other people can use it. And also something that you would like to do, but you cannot. I would like to collect more use cases. As you could see, I have only provided this boring synthetic example. So ideally, if you start using this for any image analysis workflow that you have, get in contact and let me know, because maybe we can collaborate. And also, ideally, if you have some example biological open data that you would be willing to share, that would be awesome. And that could also be part of some sample data from the plugin. So of course, I want to assess performance. So here we don't have many signals, maybe with more. I'd like to check that. And usability. So again, feedback like, oh no, I don't like this visualization and stuff. And interoperability would be also interesting. For example, you may using this plugin to do selection of signals for some purpose I did not think of. So, but I'd like it to work with whatever you're doing. So that's what I put it here. And also, of course, try to implement these enhancements for our next release. I can already give you a few, at least something I've been working with, of course, to use this tool. So it's been for decades now, of course, that there's been classification of called biosignals. So this is an example of some ECG data. And then, of course, in somebody suffering from arrhythmia, the signal is very different from somebody with a normal rhythm. And then there are medical cases where it could have the ground truth because you know the patient's conditions, and then you can use that to train a classifier and further then predict with new data. For that, you would typically measure some signal features. So there typically can be understood as signal statistics, but there could be some not very traditional statistics. And then those could be used as features for classifiers, for example. So I've been working in parallel with this other plugin called the napari-signal-selector. So this one, unfortunately, is not already released. I'm still working on it. But the idea is that it would work in conjunction with this signal selector. So it could select some signals and then run predictions on data that was not annotated before. So then I reached the end of my talk today. I'd like to thank a lot my team, as you can see here on the picture to the right. Also, I would like to thank there's some open source tools that basically build the foundation that allowed me to produce this. So napari and also napari-matplotlib, which is, as I said, the plugin I'm using to build this, and napari second image region props, which also I use some features of. And last but not least, there is the link here to the plugin, which I already sent in the chat. So that was my idea here today. It's more like a showcase of this first release, and I did not prepare any exercises. So the exercise would be try to install it, see if the instructions are clear, and give me feedback if you can. And I'm open for questions now. I think we have some, like around 8 to 10 minutes for questions, if you have some. Please open your microphone or write something on the chat. Thank you. Thanks, Alex. Thank you, Luis. Hi, Ved. Okay, so Ved asked a question. Thanks, Augustine. Okay, go for the Ved Sharma first. How does this plugin compare with StructMate? It doesn't, because the idea here is not to do tracking, although I could see maybe you could try to classify some tracking data with it. But the idea of this plugin, it's not doing segmentation and not doing tracking. You could see it if you do tracking as a, let's say, a post-processing step where you want to further classify maybe different tracks. That will depend a bit if you can provide your tracking data in the structure that this plugin accepts. And then if you can do that, it should be useful. I hope that explains. Otherwise, please raise the question again. So there's a question. Oh, Augustine, does the feature table update if I were to modify a label? Okay. If the table the table updates itself, if you modify a label on the fly. Good question. Good idea. No. So I would say, you can always provide like update the table again, or provide a new labels layer with the updated table. So for now, it doesn't update it automatically, but that's a good idea. Thanks. Thank you. Are also scatter plots possible? So in this case, no, because the idea here, you can see some markers, right? But the idea here, since it's plotting signals, it only provides like line plots because it expects this ordered manner of data. And if you want scatter, I would say go already for, well, there are others, but you could go already for napari-matplotlib, which already has a feature scatter plotter there. So if you provide the table in a similar fashion with napari-matplotlib installed, there should be a way that you can have your scatter plot. And in my plot slider, in my plotter slides, I try to, again, I'm sorry if I forgot any plotter, but I try to, yes, here, to basically show some of the plotters I've seen so far. And you can see there are other options there. All right, I would just, I think, stop recording now. And if you have further questions, I can still answer in a couple of minutes, if you prefer.